I'll be talking to you a little bit about a very active area of research here, which is managing immunotherapy through metabolism. Melanoma has been a key target for a lot of different immune-mediated treatment approaches, as you know. And one of the many reasons for this is that it's a highly mutated tumor. So compared to essentially every other known human malignancy, melanoma carries more mutations. And this is from the UV burden. This is from the UV causing a high mutation burden. And this is one of the things that makes it a particularly good model for immunotherapy. And so this is why we have led the charge in this disease. The way that the immune system is supposed to work is that when it sees a tumor, it should recognize it as other or abnormal. And the T cells, which are a kind of immune cell that are basically built to kill any abnormal cells, are supposed to come in and kill off the tumor cell. Clearly, this doesn't happen. That's how malignancies develop and get out of hand. One of the reasons for this is because tumors are sneaky and awful and intelligent. They express a marker on their surface that's called PD-L1. And the PD-L1 sees PD-1 that is expressed on T cells. And when these two bind to each other, the T cell is killed. So even if T cells manage to make it close to the tumor or into the tumor, they are then killed off by the tumor itself. The way that drugs like Keytruda or pembrolizumab and nivolumab work is by sitting at this junction between the tumor and the T cell, and thus protecting the T cell and allowing it to do what it's supposed to do, which is to kill tumor. Unquestionably, the entire treatment paradigm for advanced melanoma has changed in recent years. However, the fact remains that responses only occur in a subset of patients. And so if we think about our very first generation of immunotherapy, ipilimumab, about 10% of patients will see shrinkage of their tumors on that therapy. The newer drugs that we have in our arsenal, like pembrolizumab and nivolumab, will yield shrinkage of tumors in about 30 to 40% of patients. When we combine the two, we see responses of over 50%, but that's with a lot of toxicity, as I'm sure anyone who's received these two drugs in combination could attest to. And so many patients are not benefiting from these therapies. So what I and my colleagues spend all of our time thinking about is, why does immunotherapy work in some patients and not others? Why does it stop working in some patients for whom it did initially work? So the first set of scans might look good, but the subsequent scan might look bad. Why is that? What's changed? How can we make more patients respond to immunotherapy? How can we take that 30 to 40% and bring it way up? And how can we resensitize those patients for whom the drugs initially worked and then stopped working? And so we all look at this from a variety of different angles, and the angle I'm going to talk to you about today is metabolism. In a normal tissue that is not involved by cancer, immune cells are basking in the glow of a lot of good stuff, like high glucose, neutral pH, abundance of oxygen, readily available fatty acids, and a lot of protein. And with all of these nutrients, immune cells can do what they're supposed to do. Cancer cells, however, gobble everything up. And so what happens is that you have low glucose concentration, you have an acidic microenvironment, and when I say microenvironment, I mean the environment surrounding and within the tumor, low oxygen levels, unusable fats, and low protein. And so, as I said, the tumor cells are gobbling everything up, and so even if the T cells manage to make it in there, even if they bypass the million hurdles that tumor cells are putting up, PD-L1 is just an example I gave you, but there are many, even if the T cells manage to make it into the tumor, they have nothing to work with. They're functioning at a metabolic disadvantage. One of the keys here is to understand the concept of tumor heterogeneity, and that's just a long-winded way of saying that no two tumors are the same. So if we look at the scan of a single patient with multiple different tumors, I can assure you that if we put a needle in every single one, they're all biologically different. Tumors are different. So this is true within a patient, but also between patients. a conversation for another day is the fact that a single tumor is different in different areas. And I say that just to give you a sense of the degree of this heterogeneity. So the question we asked ourselves a few years ago now is, do different tumors have different metabolism? So what we've done is we've developed a pretty detailed platform to metabolically profile tumors as we biopsy them fresh. And what we do is we run them to the lab and we purify them to separate the immune cells from the tumor cells. And I'm going to show you first what happened when we looked at the tumor cells themselves. So this is just tumor cells. We find that tumor cells from different patients behave differently. Some of them have high oxidative metabolism. That means that they're utilizing oxygen to make energy. Some of them have high glycolytic metabolism. That means they're using glucose to make energy. Some tumors are using both methods of making energy, and some tumors are using neither. And so, yes, there's a striking difference between different patients. Tumors have different metabolism. The next question we asked is, what if we look at the T cells that we separated out from within those tumors? So you remember that figure I showed you, there's tumors and there's the immune cells in there. When we look at the T cells themselves, we find, by and large, that the metabolism of the tumor that T cell is coming from impacts the T cell. So depending on the kind of environment that the T cell came out of, it will be more or less functional. I won't put you to sleep with all the details, but suffice it to say that the immune cells that are coming from the highly oxidative tumors have this phenotype of what we call exhausted T cells. They're carrying all these labels to say I'm exhausted and I can't do what I'm supposed to do. I am a dysfunctional T cell, which is really the problem in malignancy. When we did some more detailed work to see if these T cells are secreting the stuff they're supposed to secrete to do their job, the answer is no. The T cells that are coming from the highly oxidative tumors are unable to make what they need in order to kill tumor. And so basically what I'm saying is that high oxidative metabolism, we think, is bad. So the next question we asked is, does the metabolism of an individual patient's tumor potentially predict whether or not they might respond to immunotherapy, specifically Pembro or Nivo, because we use these so frequently? And we found, indeed, the patients who have high oxidative metabolism, that's the gray line here, have a shorter duration of response. So even if it works for less time. In addition, patients whose tumors have high oxidative metabolism will progress more quickly than patients who don't have high oxidative metabolism. The golden ticket in oncology is survival, as I'm sure you all know. So the next question became, does the metabolism of the tumor impact survival? And the answer was yes. Patients who have higher oxidative metabolism have decreased survival. So we asked ourselves, if we go back to mice and we take away their ability to utilize oxidative metabolism, are they going to be more sensitive to immunotherapy? And like a lot of things in science, we go back to animal models before we try to do things in people. And so in this mouse model, we essentially did a whole lot of maneuvering to take away their ability to utilize oxidative metabolism. And lo and behold, when we did that, those mice, were much more likely to respond to immunotherapy. So how can we make the tumor microenvironment less hostile and more likely to respond to immunotherapy? You certainly can't take away the ability of a human being to utilize oxidative metabolism. So we had to find another way of doing this. So if one of the problems is that the tumor cells are using up all the oxygen, and this is a bad thing for the immune cells, how can we decrease the oxygen consumption of tumor cells? And so one of the ways we're trying to do this is by using a very commonly utilized drug, metformin, which is an oral diabetes drug. There's a lot of literature that suggests from an enormous series of patients that metformin is associated with decreased cancer incidence, but also decreased cancer-specific mortality. And we don't really know why. So one of the great labs here at the institution has been working on metformin for a while, and they have looked at whether or not metformin can impact the ability of cancer cells to use up less oxygen. And in fact, if you plate cancer cells in the lab and you sit them with metformin, they will have lower oxidative metabolism. And then if you give mice that have tumor metformin in their water, their oxidative metabolism goes down. And so we started to think that metformin might be a way in human beings of decreasing oxidative metabolism. In these same mice that I just showed you, when we treated them with metformin and immunotherapy, they were a lot more likely to respond to cancer treatment than if they received immunotherapy alone or metformin alone. So mice that get the combo do better in terms of cancer than mice that get either agent alone. So what we do here at the institution is to take things from the lab to the bedside and back. And so we've developed a clinical trial that will test this hypothesis in people. And so what we're doing is enrolling patients with advanced unresectable melanoma to either get pembrolizumab, which is oftentimes what patients get anyway at this stage of disease, or pembrolizumab and metformin. And patients are biopsied up front, and then there are a series of other biopsies that happen throughout the study. Our hypothesis is that combining the metformin with the pembrolizumab is going to increase T cell activation, which is the immune cells, and their ability to divide and their ability to kill off cancer cells, compared to T cells from patients who only got the pembrolizumab. So the different thing about this study, if you will, is that all of the questions we're asking, we answer in the lab. We don't answer them through scans or anything like that. And with support of funding from the Department of Defense, we have expanded the questions we're asking of this study, and we'll be evaluating the impact of the combination of pembrolizumab and metformin on a bunch of other immune cell types in and around the tumor, and looking at their metabolism as well and the metabolism of the entire environment. We will also be looking at the impact of the combination on the genetic landscape of immune cells. So there was a question that came up earlier about does immunotherapy change the immune system? And so that's one of the things we're asking here. So in conclusion, immunotherapy has revolutionized the treatment of advanced melanoma. However, only a subset of patients benefit. We think that metabolism may be one of the key mechanisms of resistance, and modulating the tumor metabolism might increase the benefit of immunotherapy for more patients. So we've enrolled a handful of patients on this study. It's ongoing, and I really look forward to updating you, hopefully, at the next symposium. Thank you, and I'm happy to take any questions.